Hello and welcome to another episode of Momento Malum today. In this episode we'll cover the differences between quantitative and qualitative genetic traits. The difference between these traits are most stark in the framework of disease resistance, and it also helps that this topic is one of my favorites. The first and easiest difference between quantitative and qualitative traits are that quantitative traits are genetic traits controlled by many genes, while qualitative traits are often controlled by a small number of genes, typically one gene. Quantitative disease resistance increases the general durability of the host to a specific disease by increasing the amount of natural defenses that slow down or limit the growth of the pathogen. Qualitative traits are usually controlled by genes called R genes, which provides complete immunity to a strain or specific group of strains of a specific disease. The R gene does this by triggering host cell death when a specific pathogen strain with a specific protein is detected. Once a protein is detected, the cell self-destruct button is pressed and the infected cell is killed, taking down the pathogen with it. There are two predominant categories of diseases in plants, biotrophic and necrotrophic. The former includes diseases like mildew and rust. These pathogenic fungi require live cells to survive, and the population of pathogens will do what it can to keep the host alive, so that the host does not take it with it. Necrotroph diseases are diseases that can feed on both living and dead tissue. As such, these fungal or bacterial species have no qualms in killing the host. On the botanical pathogen spectrum, fusarium and white mold are the most common diseases within this category, at least in an agricultural setting. In the context of disease resistance, specifically for biotrophic fungi and bacteria, both quantitative and qualitative traits are available within the crop genome for use in breeding projects and in common and commercial cultivars. Since necrotroph pathogens can survive on both living and dead tissue, our genes are not effective against it. As a consequence, the plant only has quantitative resistance to lean on, especially for diseases like white mold or fusarium. I've made a video on the topic of white mold resistance, link in the description below, but the too long didn't read of this video is that with quantitative resistance you'll get varying levels of resistance ranging between 0 to 99%, and this type of resistance is dependent on the environment, which means that the resistance response will change for better or for worse depending on the environment surrounding the plant. Hello, I just want to let you know, if you want to fund this channel, you can do so on the platforms seen in the description below. The main strength of qualitative disease resistance is that it works 100% of the time until it doesn't. The best way to explain this is with an example. The wheat cultivar Ceres was resistant to the majority of the wheat rust strains within North Dakota. Ceres was put into production in 1926, and its R gene prevented rust infection until 1935, when rust strain 56 evolved and overcame this cultivar. Single gene traits are in general easily overcome through mutation and natural selection, and often only require one mutation. As a consequence, R gene traits have a short shelf life. The same thing applies across all single gene resistance genes across all crops. Quantitative disease resistance has the benefit of long-term durability, but this type also has its fair share of drawbacks. For one, it's dependent on the stress level of the plant, meaning it's environment dependent, and if a strain is particularly aggressive enough, either due to genetics or environment, it will also overcome that quantitative resistance. The second drawback is that it has a hard limit of 99% resistance. You'll never get 100% resistance, you'll get close to 100%, but never 100%. And the third drawback, it's hard to breed quantitative resistance into a cultivar in comparison to breeding qualitative resistance, mainly because it's easier to breed in one gene than multiple genes. In the context of annual crop species like soybean, the infrastructure is in place for a new cultivar to be released every year, which is the case within Ontario. As a result, the ever-increasing drive for greater improvement in yield and other key traits means there's a rapid turnover in annual in annual crop cultivars. This means that the weaknesses of an R gene are less relevant in annual crops than they are in perennial crops, as the cultivar that's mainstream in one year may be outmoded by the next year. For annual crops like apples, the cultivation of such species are a longer-term investment. In Ontario, an apple orchard can last 30 years or longer before having to be replaced. Diseases like scab can be controlled by R genes. The VR gene from Malus floribunda has been incorporated into many cultivars in the late 80s and early 90s. However, as early as 1995, there is evidence of the gene breaking down in France, and several other instances of resistance breakdown have been seen across Europe. These started to appear in the 90s and early 2000s. If you compare this to the resistance of golden harvey, this cultivar was grown in the 14th century, up until the 19th century when it fell out of favor. It was well liked for its flavor and disease resistance, and its cultivar is still durable against scab, thanks to its strong quantitative resistance. As a consequence, I would argue that R gene should be a secondary resistance in comparison to strong, durable quantitative resistance, as the latter trait is built to last, and I argue that longevity should take priority above complete immunity. An indefinite durability of resistance, even if it's only partial resistance, is better than the perfect resistance that breaks down before you get what you paid for out of the orchard. But that's just my 2 cents on the matter. But that about covers everything. Thank you for watching.